Okay, I am deep into bacteria, biology, and cellular actions. Now, they're talking about cells. Now listen to this. The most life on Earth is dormant, just sleeping, after pulling an emergency brake. Now listen to what they're saying here. Many microbes and cells are in deep sleep, waiting for the right moment to activate. Biologists discovered a widespread protein that abruptly shuts down a cell's activity and turns it back on just as fast. Well, what does microbes and cells are in deep sleep waiting for the right moment to activate? Activate what? What are they going to activate? Well, they activate and they make proteins. Well, this is a cell. This is what a cell looks like inside. There's all kinds of little organelles inside of a cell. It's a single cell, but it's got a membrane around it. It's got all kinds of, there's literally millions of particles of things doing things inside of every single cell. And every single cell has all of your DNA in it. And that's the program that runs your life. Now, some cells, the program shut off and they say, hey, don't use that right now. We don't need it. Just shut it off and just wait. Stick around. We'll be back to you when we need that stuff. These proteins or enzymes. Enzymes are proteins, same things. Now, when the going gets tough, many microbes go dormant. New research discovered a ubiquitous, which means it's everywhere, protein that shuts down a cell's protein production in a flash. Turn off, turn on. It can do that. Now, why would it do that? They're saying it's doing it because we have to protect ourselves from, well, here's what they say. They say researchers recently reported discovery of a natural protein, they call it ballon, that can bring a cell's production of new proteins, which are enzymes, to a screeching halt. No more enzymes for you. Ballon was found in bacteria that hibernate in Arctic permafrost. Well, yes, it's going to be everywhere, so obviously it's going to be there. It also seems to be made by many other organisms and may be an overlooked mechanism for dormancy throughout the tree of life. Yes, every cell doesn't continuously put out the same stuff over and over all day. It's only when you need it. When you need a certain enzyme to break down a certain food, it gets produced. If you don't need it, it goes to sleep. If you get invaded by a certain bacteria, you have a program in you that says, whoa, I know what that bacteria is. Turn on XYZ over here, and it methylates, and all comes a protein that kills the bacteria or kills the invader. Now, they're saying most forms of life, the ability to shut oneself off is a vital part of staying alive. And they're talking about harsh conditions, lack of food, lack of weather, and all that. No, let me show you what happens with methylation. And I believe that's all that they have discovered is, hopefully, maybe they have discovered this ballon, which is the key to turning on and off genes. But I say it's the key to methylation. Okay, my friends, another shocking day with the discoveries that are being made about the human biome, about the process that the body uses to do its work. And there's a lot of work done, and there's a lot of different chemistry done in your body. I mean, it's just incredible the amount of chemistry. And what that requires is programs, literally programs. Your genes, when they talk about a gene or genetics, that is a program. It's literally program. And if that program is missing a couple of these it doesn't work. If it has a couple of extra it doesn't work. Now, they just came up with this big study now about how genes can turn on and off. And they're calling it a new molecule or something. I've looked into this a little bit, as much as I can. It's not detailed enough to give me what I really need to know, but I think they're just looking at methylation. And they just apparently don't understand methylation, but I could be wrong. So that's my take on it at this moment. Now let's go through this. I'll explain what they are proposing, and I'll explain what methylation is, and we'll take it from there. And like I said, I've been studying this deeply, I mean deeply, about cancer, about Alzheimer's, about autism, about all these different diseases that are sort of invasive and they just take over a person's life because they're not producing the enzymes that do the chemistry to protect you. Now, methylation is a chemical reaction in the body which a small molecule, a tiny little molecule called a methyl group, gets added to the DNA. And don't forget, in every single cell you have your DNA. So the methyl group gets added to that DNA. Proteins or other molecules. So it adds into the DNA or the proteins or other molecules, but it's in every cell. The addition of methyl groups can affect how some molecules act in the body. For example, methylation of the DNA sequence of a gene, which is the program, so the methylation of the DNA sequence of that program in that gene may turn the gene off. So it does not make a protein. It doesn't do the job of making, really the protein is an enzyme. Changes in the methylation pattern of genes or proteins can affect a person's risk of developing a disease such as cancer. I say the reason these genes don't turn on and off is because you've destroyed some of the, hold on a second. All right, there's a lot of ways you can destroy bacteria, but bacteria create all of the enzymes. They create 100% of the enzymes. As far as I'm concerned, methylation is due to enzymes. Now, basic mechanisms of DNA methylation. That's the DNA. That tells the program what to do. And the unique landscape of the DNA methylome in metal-induced carcinogenesis. What was that? That's you get cancer from certain heavy metals. And I can agree with that because they interfere with the DNA sequence. DNA methylation plays an intricate role in the regulation of gene expression. So your genes are working perfect. But if your methylation isn't there, it doesn't turn it on. If the methylation is there, it won't turn it off. So that's the important part here. And it says events that compromise the integrity of the methylome may potentially contribute to disease development. And yes, if you can't turn on the enzyme to attack the invader, you're going to be invaded. I've been talking about this for years now, and we have some studies underway right now overseas that they're looking into the biome. They're looking at it everywhere now because they realize, you see everywhere you see ads for probiotics. Probiotics is what you need. You need certain biotic they're little creatures, really. They're live, living cells. And when those cells produce a ribosome, that ribosome opens up and delivers the enzymes where they're needed. If you don't have the ribosome, you can't do it. And the only way you get the ribosome is to have the enzyme. I mean, to have the bacteria, because the ribosome holds the enzyme. So the bacteria produces the ribosome. The ribosome goes out and pops open, and all of this fancy little chemistry happens and does the job it's supposed to do. If it never methylates, it never turns on. So I think that's what these people have found, and they're making a big deal out of it. But I don't think it's what they're thinking it is. They're thinking it's something new. It's something. It's everywhere. They say it's in every single cell. Basically, they're finding. It's not like it was unique to something that's hibernating. See this? Earlier this year, publishing in Nature, researchers reported the discovery of a new hibernation factor, which they have named Balon. The protein is shockingly common. It's everywhere. It's methylation. This is really incredible. They realize it stuffs itself into every ribosome in the cell. Every ribosome has the ability to turn on and off. That's methylation. Okay, in that article, they talked about turning on and off protein production. Well, enzymes and proteins are basically the same thing. They're chemistry sets. However, proteins are normally building blocks. They build things. They grow your bones and so forth. Enzymes break down things. So there's a builder and a breaker. Now the enzymes, what they do is when you eat different foods, you need different enzymes to turn on to create the chemistry to break down the carbohydrates, the fats, so forth. Now, how does that happen? How do you get that enzyme? And does it turn on instantly when you need it? This I'm not certain of, but I believe it does. Now, these are what they call the trigger ribosomes. And these little tiny packages come in and they go to where they're needed to break down something or to build something, and then they open up the sheath and then they turn into the product. Now, let's see this as a biological action. In my model of the world, this is the most important thing there is, bacteria. Now, this is what creates those ribosomes that we're talking about that make the proteins and the enzymes. And the enzymes are catalysts. You have to have them. You don't have them, you're just going to die. You have to have a good microbiome, basically, creating the right ribosomes, which is the right chemistry to do the right job. Now, this is a DNA strand. You see it? Nucleoid DNA. This is that bacteria's DNA. You have DNA. Bacteria have DNA too. The DNA is a building program, first of all, to construct this, and what's the flagella going to look like, and what's the little this and that, and how's it going to be made. That's part of this package in the DNA. Now, additionally, there's a package in here that says, I am a producer of ribosomes. And these are the little ribosomes, and they're going to be exactly like this. When they unsheath, originally they're a little tiny ball. And when they get to where they are needed, the sheath comes off, and that's what pops out. And that's an attack dog in the case of an enzyme. In the case of a protein, it goes over and lobs onto a piece of bone or something and starts constructing the bone or tendon or whatever it is. Proteins are building blocks. This comes over and destroys something. That's as simple as I can make it. All right, so you saw that the bacteria creates the ribosome. The ribosome is that little ball. It unsheaths, you get the enzymes or proteins. And they look like this. And they are so exotic, it's just unbelievable. Now, how does that actually all happen in one sequence? Can we see it all at once? Yes. Let's take a look at it right up here. All right, here's the whole scenario, one shot. There's your bacteria. Remember, it has its own DNA, and that DNA says, I'm going to make a ribosome, and this is the ribosome. Now, how does that little ribosome turn into something you can use in your DNA? Well, first of all, those little balls are program codes. There's 20 different codes. They're amino acids. And those 20 different codes come up and they transfer this sequence of molecules. And the sequence is the program. If you take that yellow one out, it's not going to work. You take that pink one out, it's not going to work. They have to be all of those sequence, and they go into this messenger RNA, and it's a little one strand of RNA, of nucleic acid. Normally there's two strands, and they roll around together. So this one strand comes up, and it has to have something that will break in to your DNA. You see this cloud around here? There's a cloud enzyme that comes in there, and it says, okay, open up. We got the messenger RNA coming. We got to make room for them. It comes in, and it programs this chunk, which was normally programmed in a certain way, and it reprograms it. And now it's built into your DNA, and it somehow, and I'm not exactly sure how, because it's got to break into only one DNA at a time. It can't break into all of them at once. So somehow that DNA transfers this code to the rest of your DNA, because all of your DNA should be the same, no matter what cell it's in. It's got that same program. It says, and you only use certain parts of the program. The rest is methylated. It's dormant. It's sitting around waiting. It's not being used. I think that's all they found was the methylation. But this is the process of turning just stuff into a ribosome, because the bacteria literally builds the stuff. All right, that ribosome just didn't happen by itself. The bacteria somehow in their program says, take that little molecule, this one, add it over, and it puts it in here, and then it comes out, and then it does all its job. This guy here, that's the key. These are the key to evolution, is bacteria. This is what we should be studying. We should have a bacteria database. We don't have it now. We don't know what bacteria is supposed to be where. We don't know what it does. It really is this is health is right there. And this lies in this zone called the fluid-filled highway, primarily in your microbiome, in your gut. But from there, these enzymes and bacteria can go through the body through that fluid-filled highway and deliver these packages, which are these enzymes. As far as I'm concerned. Now, there's a couple other options that I've been thinking about too, because of this movement of DNA through the body. Somehow it's moving around. You program it at one place, and then it's getting to the rest of your body. Somehow it's got to be in. If they're right, all your DNA is the same, and they may not be right. You may have different DNA in different areas, but it's only going to be these sequential parts that say, I'm in the pancreas, I use this sequence here. And nobody else in the whole body has this sequence. It could be totally missing. I don't think they know, because these DNAs are so exotic. It's unbelievable the amount of program that's in here. Plus, they're working with 20 different amino acids. So you got instead of base code, ones and zeros, you got 20 different codes. The program is just way off the charts, incredible. And just think about biology. It's it just knocks me to the floor every time I even let my mind go into that realm of how could this possibly happen? How could this possibly have happened? This is all right. I got to go to the floor. No, I'm telling you, it floors me. This is what a cell looks like. This is an animal cell. Inside of this cell, remember I talked about the ribosomes? There's the ribosomes right here. The lysosomes are right here. The lysosomes slice open the ribosomes. The ribosomes come around and do the chemistry. Simple as that. Not simple, but that's what it is. Now, this is in every single cell. You have all these organelles. You have all of this different. It's just too much. Too much. There's no way ever, zero, that just somehow slime created this. A few little. It's just crazy. Who created it? I don't know, but I'm going to tell you something. Somebody that was a programmer, an engineer, a biologist, a chemist, all of that stuff. And as far as I'm concerned, the bacteria are the one that they're the most intelligent things in the human body. It's a bacteria. You don't have the bacteria, not a thing in you is going to work. Nothing will work. You'll be invaded, because these enzymes protect you in a zone right here. This zone right here. That's the protection zone. You see that? This little layer right here is this layer here. And what does that layer do? That's a membrane. See a membrane? It says you stay out of here and you stay in there. All the good stuff is down in here. And what does that look like inside? It looks like this. All right, so this is the membrane. It's got a bunch of holes. It lets stuff in and lets stuff out. It's got to. It lets good stuff in and it sends the garbage out. Absolutely. But it has all this other stuff in here that deals with the fluids. It deals with the contents that are being, whatever coming in. It has to break it down, or has to work with it in some manner or another. It has to bring it somewhere, and whatever it does. It's so complicated that it would take me weeks just to go over the organelles that are in a cell. And then when you start going into the structural part of the body and how things anchor, the things that I'm discovering, it's just off the charts crazy. Because I work with what's called mudfossils, and they are actual perfectly preserved bones and organs and everything else, and I can see all of these different layers and everything in them, because the things I'm working with are very, very large, and you can see a lot of this structure inside. I mean, right up close, and understand what you're looking at. So this is a new day in fossils, for sure, and in biology and chemistry and everything. Everything changed once I discovered the mud fossils. I realized all of these things they were talking about needed to be re-examined, and boy, I'll tell you, the re-examination is amazing. Now, this is how you get cancer. It breaks through the membrane, all the way down, which, well, it's skin, really. But skin, you can call it a membrane. And it breaks down through, but once it hits this layer, that's the fluid-filled highway. Once it hits there and it breaks through, then you're in trouble. Now, if you get a gash, this is like a growing down, stage 1, 2, 3, 4, and it just keeps growing and growing and growing. Now, you could get a gash and get invaded by all kind of bacteria down there, yes. That doesn't cause cancer necessarily. It could really be a problem. But what you're supposed to have is all the fluid-filled highways supposed to bring all the troops down here. I don't care how many bad guys you got. You got much more fluid-filled highway than they have. If you've got the right bacteria, you should be able, and creating the right enzymes, it should come down here and kill these invaders. Sooner or later, you're going to kill them because you have the right stuff. But if you don't have the right stuff in here, you're done. It's that invasion is going to kill you. Now, tumor is a different story. That just grows and grows and grows, but there's nothing to stop it, and it just keeps going. And now it gets into the vital area, and then it spreads because it can get inside your fluid-filled highway, just like the rest of your bacteria, and go find another place that's vulnerable, and that one's done. Okay, this was sent to me by my friend Pedro today, and I sent back to him saying it was an interesting article, and they extracted these cold shocked bacteria ribosomes, and then they go on and so forth. I think it's methylation. And then I talk about the chemical reaction in the body in which a small molecule, a methyl group, gets added to the DNA, proteins or other molecules. The addition of this methyl group can affect how molecules act in the body. For example, methylation of DNA sequence of a gene may turn the gene off so it does not make a protein. Let's just turn it off. Changes in the methylation patterns of genes or proteins can affect a person's risk of developing a disease such as cancer. And they turn on and off because they don't have the enzymes. That's my take. Methylation goes both ways, on and off. One way a cell can turn on and off genes is through DNA methylation. That's not my words. That's DNA methylation, is an epigenetic mark that labels the chromatin open state or off closed state. That's all. It says, okay, you're open to do your job now, or just don't go out there. Something like that. Anyway, again, I love you all. And thank you, Pedro. I appreciate it, my friend. He sends me a lot of stuff. Nice guy. All right, Pedro, thank you, buddy. This is absolutely incredible. The timing on this is amazing. I'm just getting ready to upload the video. And this comes in. Algae microbots transform lung cancer treatments. These are nothing more than little bacteria, basically. And they loaded them up with, it sounds to me like the enzymes that can kill the bacteria. And they coated them with red blood cell plasma membranes. And then they just sent them through the body with their little flagella, just like all the rest of them do. And they went down there, and See, typically, synthetic microbots are typically made of rigid metallic polymeric structures, difficult to manufacture, unable to access certain organs and tissues that can be actually toxic. Now, microalgae is just nothing more than little bugs. It overcomes these concerns. For one, microalgae can move autonomously because they got flagella. They're just like a bacteria. They propel themselves through the organs such as the lungs. They go through that fluid-filled highway. They're less toxic compared with other microorganisms. They are also cheaper and easier to produce. It sounds to me like they're creating living, well, anyway, I don't want to make statements that aren't true, but I'm trying to understand this. These hybrid microbot called algae robots combines microscopic live green microalgae, all right, commonly used in pharmaceuticals, Chlamydonis reddenhardt. They combine that with nanoparticles, little tiny, nano means like billionths of an inch, coated with red blood cell membranes. So, to me, it sounds like they're taking enzymes, coating them with red blood cell membranes so that the contents of the enzyme is in there. And then when it gets to where it has to go, they say it's this camouflage. And it prevents it from being attacked by the patient's immune system. They see it as its own blood cells. Within the nanoparticles is a common type of chemotherapy drug called doxorubicin. Now, is that an enzyme? I'll bet you that's an enzyme. Okay, final word. I give no medical information or no health advice. No medical advice. I just do research. So please don't take this as do this or do that. I just do research. I am focusing on the bacterial products, which are the enzymes. And the enzymes do the chemistry in your body because they're catalysts. And they end up affecting your DNA or breaking down food or all that stuff. So to me, these are the important guys. Now, what do we need to learn? We need to learn what's in us. And is it different in different parts of our body? And I say it could be or it may not be. But I can tell you one thing for certain. Absolutely no question whatsoever. The membrane that surrounds your liver is not going to be anything near the membrane that surrounds your lung. Your lung has to do this all day long. It's going to have all kind of fabrics in it, and it's going to have all kinds of chemistry in there to fight off these particles of air that's coming into you, because you're breathing air in, and so it's got to stop. You got to incorporate that air into your body, but you don't want to take in that bacteria. So your first line of defense is your lung tissue. All right, and it's the same as this. It's nothing more than that. It's just another membrane, and this stuff hits it and it kills it, or it just goes out as phlegm or fluid or whatever it is. But if it hits here and sticks and goes through, then you get sick. So that's your lung. Kidney's a different thing. Your liver's a different thing. They just lay there, but they're all kinds of heavy metals and everything get processed in there. And like your kidney, let's say you got salts and your acids here and there. This membrane is going to be different there than it was in your lungs. This is no question about it. It has to be. It's dealing with different chemistry. Different chemistry means there's going to be something different about that than the one that's around your heart or your whatever it is, your eyeballs. Everything's got membranes. There's nothing in you that doesn't have a membrane. So I say we should put our resources into understanding the bacteria that create the enzymes. And they will float around through the fluid-filled highway through your body and do the chemistry instead of all the other things, interventions we're doing right now. And a lot of that stuff is just not working. And I have personal experiences of this, and I would like to see this looked at in much more great detail, this bacteria, because I'll tell you, as far as I'm concerned, the bacteria are the key to the enzymes Enzymes are the key to health. They make things, and they dissolve things for your food, and to break down things to make, products available, so proteins can use them to make things grow. It's a very, very complicated set of particles doing separate little jobs. And these are the guys. All right? Thank you. I love you all.